Hello, this will be my YouTube video for the damage of UVA light on yeast lab by me, Devin Seal. General overview of this lab is that I'm going to be going over the effects of light, specifically UVA light, on yeast cells and see exactly how much damage around 5-ish minutes of UVA light will deal to these cells, whether they be exposed, or the main point of this experiment is to see if they will still be damaged even if layers of protection are used. In this experiment, the specific ones that are going to be used is different ones is plastic wrap and cardboard. And we're going to see how those may change up exactly how light treats the yeast cells to see how well these specific materials protect the yeast from the sort of damage that you normally expect UVA light over time to do. Speaking of, so why actually do this experiment though? And the main reason to do this experiment is to see, well, first off, see exactly what kind of damage UVA light can actually do, because UVA light actually is one of the main components of the light that shines on us all the time. It gets through the atmosphere and it shines down on us, UVA and UVB. And so want to see exactly how dangerous UVA light is to yeast cells, because yeast cells are similar-ish to human cells, and so if it does big damage to yeast cells, it could be an important thing to consider on whether or not it would also do damage to us. And that's why we're testing what can possibly be used to protect the yeast cells, because those types of things are also important to know for human beings, because light, like UVA light, if it is going to do damage, it can do it with surprisingly low amounts of energy. So even though it might get hit by the light and not actually increase too drastically in temperature, it may still be doing large amounts of damage to the bonds, to very important bonds in the cell, that could cause large amounts of damage to it. The general hypothesis for this cell is that if you leave an exposed place of yeast to the UVA lights, then most of those colonies are going to die. But if you leave them wrapped in something like plastic wrap, then, well, a large amount of them are going to die. A decent amount of them are going to be able to survive because of that protection. And in the case of cardboard, which is very thick and not probably going to let light through, most of, if not all of the colonies are going to be fine. And the materials you're going to need to do this is 3 sterile plates, some plastic wrap, a UVA lamp, 3 yeast samples, some foil, and cardboard. And the one that was not mentioned earlier in the hypothesis is foil, which is just because foil is generally a constant in a lot of the experiments, so it wasn't really mentioned as thoroughly. And for the setup, most trials are going to be set up very similarly, and that they are essentially just the normal yeast colonies, but say for the first one it's going to be completely exposed yeast cells exposed to the UVA lights for the constant 5 minutes with the same lamp. The second trial is going to be wrapped in plastic instead, and they're going to be exposed to it. The third is going to be covered in foil, which I mentioned earlier we hadn't really gone over the hypotheses, and they'll be exposed to the light. And the fourth and the final trial is going to be the cardboard cover, which is one of the main focus ones for the kind of purpose of this. And so they're all going to be exposed to the light for 5 minutes, and we're going to see exactly what happens with their survival rates. So what happened? For the exposed, around half of those cells died, or a bit more. And for the plastic wrap, around 40% died, around 10% of foil wrap cells died, and almost no cells died left by the cardboard. As you can see, our little cardboard shield here, this guy is just fine. But the general thing to see here is that, well, in the different protections from exposed, where there really is no protection to the 5 minutes at all, for the plastic wrap it provides a little bit, and for foil and cardboard, which are the ones that are very not light penetrable at all, you can see that there is very, very few of the colonies are actually dying. And that's the kind of conclusion that we expect with the hypotheses, where it's just the stuff like cardboard and foil is very high in protection of these things because light really cannot penetrate it. And then generally for our conclusion, UVA rays have the ability to do a lot of damage to unprotected cells, even after only a few minutes of that. However, that damage is not an unavoidable danger, as things can protect them very well, which is a lot of times why we stay in shelters, because generally the most effective barrier for these types of things is very solid, dense material that light generally cannot get through to actually damage our cells consistently. One final note is that all damage is not the same. Just because a lot of light is hitting the cells in the colonies, not all of them are going to die quite as badly, I guess I'll say, where some of the cells can actually be brought back if they're treated properly after the experiment, which just shows that sometimes the damage dealt to the cell may not actually be too severe, or it might be too bad, and it shows that the damage that light can do, as it's kind of unpredictable, can vary a lot. Like, even in the exposed cells, a lot of them did survive just because light happened not to do the damage, which just shows the unpredictability of light in these situations. Just here at the end, I wanted to show all the references that were used.